Hello, students. A very good evening. So today we are at the last part of our neural control and coordination. So that is quite a little bit of an important topic. I can't stress that it is way too important for our NEET examinations, but I can stress on the topics that might occur or the questions that they might point out in asking. Okay. So, anyways, so we will talk about our sense organs. So in the last class, we discussed about human brain and the activities of human brain and the different roles that different parts of the brains undertake and how meticulously wired it is and how it enables us to perform how we perform, right? So, but it is not possible for us to achieve this kind of a consciousness without the support of our sense organs. So, the sense organs in our body plays a very prominent role, okay, in understanding many different things, isn't it? So the first thing that we have to give regards to our sense organs is in understanding the environment that we live in. So without the eyes, you would not be able to have the sight and understand where you live and who is your enemy, who is your friend, what to eat, what not to eat, etc. The same way goes with the rest of the sense organs. It might be your hearing abilities. It might be, see, you just see the hearing. So you might think that hearing just gives you the sense of sound, but uh, along with the sense of sound, it also gives you an ability to balance your body. Okay, the balance, like whether you're centered, your body is centered, or you're leaning towards left, or you're leaning towards right. So this sensation comes because with our auditory sensations. And the third thing is the tongue, the taste, the food, the variety of chemicals that dissolve in your tongue and it gives you a sensation of the taste. It might be pleasurable, it might be sometimes rejectful to our body. So it basically understand our food, okay, and the things that we consume, okay. And the biggest sense organ that we have is the skin, and you know how effective is the skin, not only as a prominent physical barrier that prevents many of these unnecessary things to enter our body, but also the touch, the pain, the pleasure, the temperature, or understanding the texture. So all the sensation, the somatosensation of the skin, okay, is one of the prominent senses. So as I've told you, we have many sense organs, such as we have smell sensation, we have sight sensation, we have touch, and we have hearing, as well as we have taste. So coming to different sense organs, the two important sense organs that we are going to discuss in detail is the sight, or the sense of sight. Okay, as simple as that. Now, first we have to understand the anatomy, and then we have to understand the physiology of to just have a brief idea about how we are able to see objects or how our visual impression is built. Okay, so we know that the organ that is responsible for our sight is our eyes. And if you see your facial bones, and our facial bones have a very beautiful sockets, which you call it as the eye sockets, in which the almost spherical shaped eyeballs are placed. So we have two eyes, obviously, right? So these are very well placed in the sockets of the skull, and they're called as the orbits of the skull, okay, or the eye orbits, okay. So as I told you, our eyeball is nearly spherical, okay. So if I take the eye and if I take a cross section of the eye, what I can observe is our eye is made up of three important layers. What are those? One is the sclera, second one is choroid, and the third one is retina. So students, you have to just uh, kind of remember more about eye. Okay. So I will stress on some of the important things that you have to memorize. So please go through it while you're revising. Okay, first thing that we have to understand is the outermost layer, that is the sclera. So if you see that what is the outermost layer that is present in your eye, that is the sclera. So it is a very dense connective tissue, okay, and it forms the external layer of our eye, okay. So if you see the sclera covers the entire eye, okay. So if you see the structure of the eye, the entire eye, you can see the blue portion that is present here. So the entire portion of the eye is covered by sclera, which is a dense connective tissue. just anterior, that is above the sclera, what do we have is two more layers, that is the choroid and the retinal layers. Okay, see here, anterior to this is the sclera, and anterior to sclera, what do we have? We have cornea. Okay, so anterior to our sclera, what do we have? We have the corneal region. Now we understood what is sclera. The second layer is choroid layer. The choroid layer is kind of you can remember choroid layer very easily. How you can remember choroid very layer very easily is you just follow this blue line. So if you follow this blue line, okay, so this blue line, what you can see here is at the anterior portion, you can see that it is quite thick. That is the 1/3 of this blue line is quite thick, correct, right? And then the posterior portions, that is the 2/3 of the posterior parts, is quite thin. So this region, which is not quite uniform in its thickness, so that is nothing but the choroid layer. And if you see the choroid layer is usually bluish layer, and it is the middle layer of our eye. Okay, so what does choroid contain? Choroid is very richly supplied with blood vessels, and as I've told you, there are almost majority of your choroid layer in the posterior region. How much of it? Almost 2/3 of your choroid, okay, is of your eyeball is actually thin region, whereas only 1/3 of it, which is anterior part, is actually thin region. Okay, the so okay, sorry, thick region. All right, sorry. 2/3 is thin region and only one part is thick region. Okay. And wherever you see the thick region, what can you see here? You can see that the choroid is coming here and it is forming a very important part. You see, a very important part, and that is nothing but your ciliary body. Okay. So what is your ciliary body? So from here, if you just understand, okay, your ciliary body, it just continues forward, okay, to form, whatever you see, the color of your eye. You can see that your eye is whitish on the sides, and then it starts blacking, and then you can differentiate the color of human eye. So that colored portion, which is also called as pigmented portion, and opaque portion, okay, which is called as iris. So what did I tell you? The thick portions forms the ciliary body, and the ciliary body, they just continue forward to form what? To the visible pigmented regions as well as your opaque portions of your eye, which is called as the iris. Now, what is the iris? You know that iris is a colored portion, and you can see that at the center of the iris, okay, you have an opening, and this is called as a pupil. Okay, so this opening of the iris, what you can see, is called as pupil. Now, sometimes what happens whenever you get very excited, whenever you're experiencing pleasure, or whenever you're, in the darkness, or whenever you go suddenly to the light, you can see that the size of the pupils actually changes. Okay, so we call it as pupil constriction or pupil dilation. Okay, he will tell his pupils are dilated. So now the pupil size can be regulated. And the reason why the pupil size can be regulated is because it's been supported by muscle fibers of the iris. So the iris muscle fibers support this pupil to change its diameter. So that is when we call it as whether the pupil can dilate or whether it cannot dilate. Okay. Now, usually, if you see our eyeball would be not a really a optical organ without the presence of a crystalline lens. So you can see that there is a prominent lens which is very transparent in nature, and it is also supported by the ligaments, okay, of the ciliary body. Okay, so the lens is very well placed in the center, and it is attached by the ligaments of the ciliary body. So this is the lens, correct, right? So this is the lens. So next one, coming to, we understood what is sclera, and then we understood what is choroid, and we understood the parts that is our choroid will give rise to, which is the iris, which is the pupil, which is the lens. Okay, and then we understood what is the ciliary body, etc. Now the third portion is the retina. So the retina is like a screen, which forms the inner layer of our eye. So if you see the retina is composed of three important cells. Okay, see, if I start from here to here, you understand this point? If I go from the inner to the outside, okay, so inside to outside, if you see how the retina layers are arranged, they are arranged with the help of three different types of cells. You have to remember the order of the cells. So this is one important part for your examination point of view. Okay, that you have to remember this. What is that? What is the arrangement of cells, or how is the orientation of the cells that are present in your retina, or how are the cells from inside to outside are arranged in the retina? So if the question are asked like this, from going from inside to the outside, what you can see is the innermost layer contains the ganglion cells, okay, and the middle layer is composed of bipolar cells, and the outermost layer is made up of photoreceptor cells. So you can see here in this picture very clearly. So these cells are called as the ganglion cells. Okay, these are called as the ganglion cells. Okay, and these cells are called as the bipolar cells. Okay, and these cells are called as photoreceptor cells. Okay, or yeah, we can call it as photoreceptor cells. Understood? So very, very, very important. Okay. Please understand this. Okay. So remember this. Okay. Now coming to your photoreceptor cells. So photoreception is basically you able to differentiate colors, light, and darkness. And the reception is very, very important. And our ability to differentiate light comes because of two important types of cells. Those are nothing but rods and cones. Okay. So now, how are they able to help you to differentiate light? They are able to help you to differentiate light is because they have something called as photosensitive proteins and pigments. They have photopigments as well as proteins that help us basically to differentiate light. Now, what are these photosynthetic pigments, or sorry, photopigments? The photopigments are basically made up of, remember this, a particular protein called as opsin and an aldehyde of vitamin A called as retinol. So you need opsin and retinol to function together to get the reception of light. Okay. So what do our photopigments are made up of? They are made up of a protein called as an opsin and an aldehyde of vitamin A called as retinol. So that's why they tell you to drink vitamin A. Okay. Sorry, milk. Okay. So anyway, on carrots. So anyways, coming to our retina, and as I told you, we have to understand what are these photosensitive cells, that is rods and cones. So first we go and understand cone cells. So if you look at the cone cells, the function of the cone cells is usually photopic function, we call it as. So what is photopic function? Photopic vision or photopic function is nothing but your ability to understand the daylight or the colors in the bright lights, I can tell you. Okay, that is your day vision is given because of the cone cells. So the function of the cone cells is to have daylight vision or photopic vision or colored vision. This is another important point you have to remember. What is that? What is photopic? Photopic is nothing but photo means what? Photon, light, correct, right? So the daylight. Okay. Now, there are actually three types of cones that are present in your photopigments, containing different types of photopigments. So these three different type of photopigments will help us understand and differentiate three different colors. What are those? Red color, green color, and blue color. You might have heard about this color blindness. Okay. Usually the person will not be able to differentiate either red color or either green color or either blue color, or sometimes red and green. Okay. And then it will be red and blue, sometimes one color, sometimes two colors, sometimes all colors. Okay. So this color blindness, the reason is the same thing. Whatever the proteins that are responsible for your photoreception, okay, which we call it as photopigments. So these photopigments will not be formed properly in the eyes, especially in the cones. Okay, so that is when a person will be not be able to differentiate the shades of red, the shades of green, and the shades of blue. You might think that I see like hundreds of different types of shades. How is it that only three colors my cones are able to differentiate? Right now, you're looking at different colors on the board, right? You can see within this thing how many colors you can see. But how is it even possible? So basically, it depends upon the wavelength and the frequency of the light that is reflecting off the object. So whenever these comes and hits our eyes, so these red, green, and blue, they act like pixels, and the combination of this is what will give you the reception of rest of the colors. Okay. So now what happens? So different colors are sensed because of the combination of these cones and their photo pigments. Understood? So what did I tell you? Cones that you one thing you have to remember is the photopic vision as or the daylight vision. Gone, done. Next one is the rod cells or the cone cells is done. Next one is the rod cells. Now here I told you it is photopic vision. Here it is called as scotopic vision. Okay. Scotopic vision is also called as the night vision. So one thing I will help you to remember, scotopic vision, the pirates, they used to put this patch on their eye, in one eye. It's not because they have lost their eyeball. One eye, they used to keep it closed because they want to increase the sensitivity of this rods so that they will be able to get this night vision. Okay, twilight vision, we call it as. Okay, so twilight vision is also called as a night vision. So basically, you're able to see at night even the dim colors. Okay, even in the darkness you're able to differentiate sometimes. That is because these rod cells, they contain a specific pigment which is purplish in color. Okay, purplish red protein, we call it as. So what is the name of this purplish red protein? It is called as rhodopsin. So the rhodopsin is the derivative of uh, you know uh, it is a derivative of vitamin A. I told it is an aldehyde of vitamin A. So that is what will give you the ability to see at night. Clear, students? Chalo. Now, coming to our retina again, what you can see is, if you see the whole of a retina, at just, at the posterior region, okay, you can see that there is a spot where all the optic nerves, all the blood vessels, okay, all the lymph tissues will start leaving the eye. So this spot where your optic nerves, okay, and then your blood vessels, they enter or leave your eye. So this place, there is no photoreceptors or there are no photoreceptor cells. So the site at which they enter or leave is also called as the blind spot. Why it is called as a blind spot? So this is called as a blind spot. So why this region is called as blind spot? Because there are no photoreceptor cells and this space is used for what? It is used for the optic nerves to leave, okay, and the blood vessels to enter and leave, okay. So this region is called as the blind spot. Clear, students? And next, just lateral to the blind spot, you see, this is our blind spot, and just lateral to the blind spot, you have one more important site. This also you have to remember very, very surely. And this spot is also called as fova centralis. Okay, this region is called as what? Fova centralis. So when I say fova centralis, this region, okay, this region is also called as macula, okay, macula lutea. It is also called as an yellow spot. So beside the blind spot, we have the yellow spot, or it is also called as fova centralis. And here you have a large amount of pigments, your photoreceptive pigments. That is, contains large amount of cones. So since it is very densely packed with cones, this region is where you have the highest sensation of vision. Okay. So lateral to the blind spot, what do we have? We have fova centralis, or it is also called as the yellow spot, or it is also called as macula lutea. So here, what do we have? We have a lot of packed photo sensitive cells, that is the cones. So that is why we have the highest resolution here. Okay. So that is when you can see the great visual accuracy or the resolution is present where it is present in the fovea region. It is also called as fovea centralis region. They have asked many times about the fovea centralis. Please try to emphasize it and remember it. Clear, students? Okay. Next one, What we can see, we understood what is cornea, what is iris, what is lens, what is retina, what is sclera, what is choroid, what is fovea, what is blind spot, everything. But one thing what we have left is there are two important spaces that are present. And the spaces are, one space is between the cornea and the lens. So what is cornea? This is our cornea and this is our lens. And this space, okay, is not empty. It is filled by a fluid. And this fluid is called as aqueous humor. Okay, what is it called as? Aqueous humor. And since this fills this something called as aqueous humor, the chamber is called as aqueous chamber. Okay. So there is a space between cornea and the lens, and this space is called as aqueous chamber. And then this chamber contains a fluid. It's not empty, it contains fluid, and this fluid is called as aqueous humor. Okay, which is a thin watery fluid. Next one. We have the second space, which is a very large space. Okay, where is this space? This is the lens, and this is the retina. What do we have? We have the space between the lens and the retina. So this retina encloses this space basically. So this space is called as vitreous chamber. So when you have vitreous chamber, you know that vitreous chamber is not empty, and vitreous chamber is also filled by this fluid called as vitreous humor. So they will ask you where is aqueous humor or aqueous chamber present. When I ask you about aqueous chamber or aqueous humor, you should know that it is between the cornea and the lens. Okay. When I ask about the vitreous humor, we should talk about the lens and the retina. Okay, the large chamber is called as vitreous humor, which is like a jelly-like substance, not watery substance, which fills this vitreous chamber. Clear? Chalo. Now we have to talk about the sensation of the vision now, or how the mechanism of the vision. 1 second, students. Okay. Yeah. How does the mechanism of vision really is? So first, what happens? We have to get the light, reflect on an object. Okay, it falls on an object, incidence on an object, I can tell. After it incidence on an object, some amount of light is absorbed by the object and some amount of it is reflected back. Whatever the light is reflected, it reaches our eyes. Okay. So whatever the light that is reflected from the object, it enters our eye. So how does it enter? The first thing, that is a transparent layer, that the first layer that it has to go through is the cornea. Okay. It goes through the cornea and it reaches the lens. Once it reaches the lens, it goes and focuses on the retina. So once it falls on the retina, we know that there are photopigments, okay, that are present in our retina, where dissociation of our two important proteins. Remember I told something called as retinol and opsin. So the retinol and opsin will be together. With the interaction with the light, what will happen? They'll dissociate. Once they dissociate, what they do is, on the amount of dissociation they do, the permeability of light through this membrane changes. So once they changes the membrane permeability, okay, see here. So the light falls through the cornea and it goes through the lens and then it comes through the and it falls on your cones and rods. So that leads to the dissociation of your retinol and opsin, and that leads to the change in membrane permeability. Once the membrane permeability changes, what happens? So they generate this permeability of this membrane generates potential differences. You know what is a role of potential differences? We saw it when we talk about neural impulse conduction, right? So this impulse that is created because of the permeability, okay, that will generate an action potential. So once the action potential is generated, where it is generated? It first generated in the ganglion cells, and then it goes through, okay, so it goes through bipolar cells, and the impulses are finally transferred to optic nerves, and then it is going to the brain, and finally, what part of the brain? To the visual cortex or the optic lobes of the brain. So these impulses are again analyzed by various parts. You know that we have associated regions as well as we have visual cortex region. So once it gets properly analyzed, so this, you see the object, you learn the object, or you recognize the object based on the memory and your experience. And finally, you have a vision. The vision is not just seeing the object, you just define the object based on memory and perception, and you build an idea around this object. So all this is happening with one side. Okay, you understood, right? Yeah. So you can see here how this photoreceptors actually they react and they change the membrane permeability. So that leads to the change in you know the potential. Which potential? The potential difference is changed in the photoreceptors that will generate action potential, okay, in ganglion cells through bipolar cells, and finally it goes to the optic nerves, and then it goes to the brain, and then it goes to our visual cortex, and then it gets associated with different regions of the brain, and finally we have the vision based on experience and memory. Clear, students? Yeah. One second. All right, so we will try to solve some questions. Okay, we have few minutes for solving, and it was very, very important for us to understand this particular topic. Okay, anyways, so we have some of them are previous year questions. I told you the emphasizing we can emphasize a very specific topic for solving the questions. So let's see. So we can understand by solving these questions which regions they might ask. Okay. So the gap between two adjacent myelin sheaths, okay, are called as what? So our options are nodes of Ranvier, and then we have axon terminal, we have synaptic knob, and then we have Schwann cells. So here are our options. So if you look at these questions, you already know that our neuron, just remember this, our neuron is very, very simple structure to remember, but not as a functional cell. Okay. So it's very complicated, cell. So anyways, so we know that our neuron has something called as, cytoplasmic extension. And one of the largest cytoplasmic extension, it comes with the axon, and the axon terminates with a branch-like structures, which is called as synaptic knob, okay, or it's called as synapses. And here, there are small minor cytoplasmic extensions, which we call it as the dendrites. Correct, right? Okay. So, and here we have a central nucleus, and then there are lots of granular-like structures. Okay. So, what are these granular-like structures? So, this granular-like structures are a special type of endoplasmic reticulum that you can see, which help in protein synthesis of your, nerve cells. So these are called as Niecel's granules. Okay, they are called as what? Niecel's granules. And then the large cytoplasmic extensions, they are surrounded by some specialized cells, which we call it as Schwann cells. And the role of Schwann cells is to, prepare what do you tell? Myelin. Okay, they secrete myelin sheath. Okay, that is a role of the Schwann cells. Now, this myelin sheath is like an insulation which prevents any of the short circuits, something like that. So, but the myelin sheath is not continuous. There are gaps between this myelin sheath, and this gaps between this myelin sheath is called as nodes of Ranvier. So the option, whenever they talk about the gaps between two adjacent myelin sheath, and we have to talk about nodes of Ranvier. Understood? Okay. And what is synaptic knob? Synaptic knob is nothing but you have the synaptic branch, okay, or it is also called as nerve end. So if you see the nerve end, there is a bulb-like structures which has vesicles, and the vesicles have neurotransmitters. Okay, and this is nothing but synaptic knob. Okay, I told you about Schwann cells. Sometimes they surround and they don't secrete myelin. Sometimes they surround and secrete myelin. So these are nothing but Schwann cells. Next one, which part of the brain is responsible for thermoregulation? These kind of questions are very, very often, okay, with respect to this chapter. So our options are medulla oblongata, cerebrum, hypothalamus, and corpus callosum. So what is your option? Whenever you talk about thermoregulation, and then your feeling of thirst, your feeling of hunger, okay, and then you're balancing your basically the sweat balance. You know, you sometimes you sweat out, sometimes you burn more fat to keep your body warm. So this is done by hypothalamus. Okay, remember this. Hypothalamus is the one that is responsible for that. I told you while I was teaching you the topics of okay what parts of the brain control what units. So I had given you a clue also how to remember hypothalamus, especially when it comes to thirst. Okay, and especially when it comes to thermoregulation. Okay, so chalo. Next one. Bipolar neurons are found in okay. Bipolar neurons are found where in humans, brain, spinal cord, embryonic stage, and then you have olfactory epithelium. So bipolar neurons are something that has two dendrites, okay, and they are responsible for transmitting the signal from two directions, like it takes from here to there and there to here. These are called as bipolar neurons. And bipolar neurons are basically present in sensory organs, especially like olfaction, that is smelling, and the vision. And then you talk about temporal, okay, auditory functions or touch functions. So here our given options, we have something that is with respect to sensation, that is the olfactory epithelium. So our option is olfaction. Okay, whereas embryonic have the unipolar neurons, and the spinal cord and the brain have multipolar neurons. So these are two multi, and this is one unipolar, and this is called as bipolar. Unipolar neurons are extremely rare. Okay, extremely rare. They are found very little in embryonic stage, and then later they disappear. Okay, so chalo. The parts of the human brain that helps in regulation of sexual behavior, yeah, okay, expression of excitement, pleasure, rage, fear, etc., are called corpus callosum and thalamus, limbic system and hypothalamus, corpora quadrigemina, and then you have hippocampus, and then you have brainstem and epithalamus. I know that these words can confuse you, but I have told you one important clue to remember. What is that? Your limbic system. The limbic system is called as emotion center, and all your behavior with respect to emotions such as sexual behavior, anger, excitement, fear, rage, pleasure, all of these things is developed in limbic system. I told you how limbic system can make you act like a monkey, right? So it controls the threads of emotions, and you act to the emotions and you become monkey. So that's why you become a master, not a monkey of limbic system. I told you this clue. Correct, right? So remember that. And whenever it comes to the expression of excitement, pleasure, rage, fear, as well as sexual behavior, you have to go for limbic system and hypothalamus. Understood, students? So this was one of the very recent previous year questions that was asked. Oh, anyways, sorry, we are out of time. We will solve further questions tomorrow while we discuss here and the rest of the topics. Okay. Thank you so much, students, for attending the session. I'll see you in tomorrow's session.